It'll take a little while for everybody to pop in. Can you guys see the attendees too? Xavier?
Sorry?
Can you see the attendees' names and stuff too?
Yeah, I can see now.
Good morning, everybody. Welcome to the webinar. We're just gonna wait a couple more minutes as people pop in. We'll start probably at 8:05 in 2 minutes. I guess it's not morning for everybody, but good afternoon, good evening. Xavier, what time is it in Switzerland? Is it 8 p.m.?
It's 5. Yeah, for me it's good afternoon.
Good afternoon. Okay, well, it looks like we have a sort of stable number of attendees, so we'll go ahead and get started. So, hi everybody, I'm Allie Boehm, a professor at Stanford, and I'd like to introduce our two fabulous speakers today, Katy Graham and Xavier Fernandez. Katy is a PhD student at Stanford and Xavier is a postdoc at EPFL. I wanted to sort of set the ground rules, but first I want to thank these two speakers as well as all the rest of you who are doing your service to the pandemic during good times and bad. So this is a challenging time for all of us to maintain our research and do this important work to try to help the response to the pandemic. This is how it'll work. There's at the bottom of your webinar screen, there's both a chat, and I see a lot of people are saying good morning, and it's nice to see you all. And then there's a Q&A button. And so we're gonna be using the Q&A for questions, and you can upvote questions. There'll be like a thumbs up. And so if you like the question or you have the same question, you should upvote it. Because then I will, Yesith, who will be joining a little late, or I will ask the questions of the speakers that sort of get upvoted. But we will not be monitoring the chat for questions. So if you have a question, please put it in the Q&A. Okay, so with that, we're gonna go ahead and get started. So Katy Graham is gonna be our first speaker, and we're gonna have her share her screen and get going. And Katy, you have 20 minutes, and if you get to 20 minutes and haven't finished, I will unmute myself and I will remind you that it's time to stop. Okay?
Sounds good. Thanks, Allie.
Okay.
All right. All right. Good morning, everyone. My name is Katy Graham. I'm a student at Stanford University in Allie Boehm's group. And today I'll be talking to you about our project in collaboration with Krista Wigginton at the University of Michigan on SARS-CoV-2 in primary settled solids and influent, a comparison and longitudinal time series in the San Francisco Bay Area. And our entire study team, because this work really took a lot of people, is listed below. As the pandemic began, many researchers in our scientific communities realized the utility of wastewater-based epidemiology in detecting COVID-19 infections in a community and as a tool to advise public health officials. Many characteristics of SARS-CoV-2 infections created difficulties in detecting cases and the spread of the virus, including asymptomatic cases and the high frequency of asymptomatic shedders that can spread the virus, lag times between patient testing and patients receiving results, allowed for the virus to spread undetected in many communities. However, it's estimated that about 50% of individuals shed the virus in their stool, which allows us to detect the virus in wastewater and potentially use the data for wastewater-based epidemiology. And this is an established concept. It's been used to detect narcotic drugs, as we saw in the previous RCN webinar, poliovirus in sewage, as well as other pathogens. but many in our community saw the utility of this approach at the beginning of the pandemic. And one advantage of this approach is that each wastewater sample is essentially a biologically composite sample, meaning many people contribute to one sample. This allows us to test for community health and trends in community health, while protecting the privacy of individuals. And in addition, it's very low effort for people to contribute to the sample. They only really need to continue their life as usual or use the bathroom. And this has been a really exciting premise. There have been a lot of really cool interviews in the media by many researchers working on this. And it's been really exciting working on a project that's so topical and relevant and interesting to non-scientists. But I do, like Ali said, want to acknowledge the difficulties of working on these projects. I think all of us have experienced supply chain issues, which can cause massive lab slowdowns. And in addition, the pandemic's effects on us all. I, probably like many people, have been personally affected by the pandemic, and dealing with grief during this time has made this really challenging work, but it's also really, really motivating work. I think it's a good reminder for me as a student that as environmental engineers, we have a duty to investigate and develop tools to help public health officials protect our communities. So in response to this call to action, our group at Stanford created SCAN, which stands for Sewer Coronavirus Alert Network, whereby we take samples from wastewater treatment plants, we process them in our lab, and then we provide our data to to public health officials. And thus far, our project has really been split into about 3 phases. Phase 1, we developed methods for wastewater treatment laboratory work. In phase 2, we created a high-frequency longitudinal time series from one wastewater treatment plant in San Jose, California. And in phase 3, which we're currently working on, we're creating longitudinal time series from multiple locations around the country. For the purpose of this webinar, I'll really only be focusing on the first 2 phases. As a part of our project, we've been in communication with 50 wastewater treatment plants from across the United States. This is a map of locations that we're receiving samples from, and the diameter of each dot is weighted by the number of wastewater treatment plants that we're receiving samples from that location. As much as possible, we're trying to collect paired influent and primary settled solid samples, and we started doing this at the beginning of the pandemic owing to our knowledge of the absorption of enveloped viruses to wastewater solids. So we thought it was a good idea to target both matrices to test which might be a more successful target for wastewater-based epidemiology. So now I'll go into some details on our methods development work. When we were developing an optimized method for influent, we compared 2 concentration techniques, PEG precipitation and ultrafiltration, which have been well cited in the literature for their ability to concentrate viruses in wastewater. We used the QIAGEN All-Prep Power Viral DNA RNA extraction kit for RNA extraction. And we compared 2 different quantification technologies, RT-qPCR and dd-RT-PCR, for quantifying N1 and N2 targets on the SARS-CoV-2 genome. And in terms of recoveries, we were using bovine coronavirus. We bought a vaccine and used that as an extraction control. We would spike that into RNA extracts prior to extraction. We were using MHV at this point as a full process recovery control, meaning we would spike that into samples prior to concentration. And in addition, we were measuring pepper mild mottled virus as a fecal strength indicator. We wanted to account for any dilution in human-derived sewage by stormwater or industrial wastewater, and we were able to use PMMOV to do that. Here are some details on the methods that we used to concentrate influent. Like I said, we compared PEG precipitation and ultrafiltration. And when we were comparing these 2 concentration methods, we would start with 1 influent sample and split it into multiple biological replicates. From these biological replicates, we would remove solids by centrifugation and retain the supernatant. For the PEG precipitation method, we added PEG 8000 and salt, mixed and incubated at 4 degrees Celsius, and then centrifuged that sample, creating a solid pellet and a liquid supernatant. We decanted the supernatant and then resuspended the pellet in PBS, and then we used that viral concentrate for RNA extraction. For the ultrafiltration method, we similarly removed solids using centrifugation and retained the supernatant. We then applied this supernatant to the top of a 100 kilodalton Amicron ultrafilter, spun this and retained the retentate as our viral concentrate for RNA extraction. The results from these experiments were that we found, in general, comparable results between PEG precipitation and ultrafiltration using RT-qPCR. In the plot below, you can see the concentration of PMMOV, and the concentration here has been log 10 transformed. And on the right, you can see the concentration of MHV. And again, the concentrations here have been log 10 transformed. On the x-axis, you can see that we've compared the concentrations attained using ultrafiltration versus PEG precipitation in terms of the concentration method. And for the blue bars, each RNA extract has been run undiluted during the RT step of a two-step RT-qPCR assay, and the cDNA has been run also undiluted during the qPCR step. And we are able to attain a slightly increased recovery in concentration for both PMMOV and MHV compared to ultrafiltration. For that reason, and in addition, because of concerns regarding the supply chain and the availability of Amicon ultrafilters, we decided to proceed with PEG precipitation. In addition, I wanted to point out the green bars, which were generated using an two-step RT-qPCR assay, where we diluted RNA 1 to 10 prior to the RT step, and then ran that resulting cDNA undiluted during the qPCR step. And we were able to achieve slightly higher concentrations using that technique, as opposed to leaving all the extracts and cDNA undiluted. So we did see some evidence of RT inhibition for this assay. We did the same thing by running RNA extracts undiluted during the RT step, and then diluting them 1 to 10, diluting the cDNA 1 to 10 during the qPCR step, and we were not able to achieve these same concentrations. In addition, we were also able to detect N1 and N2 targets in samples using ddRT-PCR that were previously non-detects using RT-qPCR. So we found that ddRT-PCR was a superior way of quantifying SARS-CoV-2 in our samples as opposed to RT-qPCR. So the overview of our optimized influent method, we chose to proceed with PEG precipitation. We're still using the all-prep power viral DNA RNA extraction kit. And we decided to proceed with ddRT-PCR as the quantification method. At this point, we eliminated MHV as a process control, and we decided to continue only using spiked in bovine coronavirus as our recovery control, and we retained PMMOV as a fecal strength indicator. In parallel, our group was also developing optimized methods for primary settled solid samples. We're comparing two concentration techniques, direct extraction of solids versus a glycine treatment procedure, two RNA extraction kits, the QIAGEN Power Fecal RNA extraction kit and the QIAGEN RNeasy Power Soil extraction kit. And similar to influent, we're comparing RT-qPCR and ddRT-PCR. Again, we were quantifying N1 and N2 in our samples, as well as bovine coronavirus and MHV as recovery controls. We would spike those in after concentration, but prior to RNA extraction. And we're also still monitoring PMMOV here. Here's some details on the primary settled solid concentration methods. When we're comparing the glycine treatment to the direct extraction technique, we would start with one sample of primary settled solids and split it again into biological replicates. For the glycine treatment, we would then centrifuge this biological replicate, which would create a solid layer and a liquid supernatant. We would pour off the supernatant and add glycine. We then would mix and incubate this, syringe filter the glycine supernatant, and retain that filtrate as our RNA, or as our viral concentrate to extract RNA using either the Power Fecal kit or the RNeasy Power Soil kit. For the direct extraction technique, we would take the biological replicate, again centrifuge it, creating a solids layer and a liquid supernatant. We'd pour off the supernatant and retain the solids layer. We would then directly extract from aliquots of that solids layer. The results from these comparisons for these primary settled solid samples were that, first of all, we were able to see that the RNeasy kit allowed for 10 times more mass per extraction than the Power Fecal kit. The RNeasy kit allows you to add up to 2 grams, whereas the Power Fecal kit allows you to add up to 0.2 grams. The RNeasy kit, therefore, allows for a lower theoretical limit of detection. In the plot here, concentrations and gene copies per gram is on the y-axis, and the dilution factor that we ran the RNA at is on the x-axis. This is using a one-step dd-RT-PCR assay. And as you can see, for each dilution, we were able to achieve slightly higher recovery, and this is for each point represents an average between N1 and N2, and each point is a separate trial. But as you can see, the gray circles for the RNA-Z data were able to achieve a slightly higher concentration in comparison to the Power Fecal kit. So for that reason, we continued using the RNA-Z Power Soil kit exclusively. In addition, we were able to see that the glycine treatment was largely unsuccessful in concentrating SARS-CoV-2 in solid samples, as well as our recovery targets. Using both quantification methods, we saw nearly all samples were non-detect. However, direct extraction of solids was more successful when we were using dd-RT-PCR rather than RT-qPCR. We were able to obtain quantifiable results using dd-RT-PCR and proceeded with using that method of quantification exclusively. So the overview of our optimized primary subtle solids method is a direct extraction of solids using the RNA-Z Power Soil kit and dd-RT-PCR. At this point, we were quantifying N1 and N2, as well as the bovine coronavirus as a recovery control, and PMMOV as a fecal strength indicator. Now that we had two optimized methods, one for influent and one for primary subtle solids, we wanted to head-to-head compare these two optimized methods in a short time series from one wastewater treatment plant in San Jose, California. And the results from this head-to-head comparison indicated that primary subtle solids were a good target for us for wastewater-based epidemiology. In the plots below, you can see that the top two plots are for the influent samples, and the bottom two plots are for the solid samples. And if a sample was a non-detect, the shape was not filled in. On the left-hand side are N1 results, and the right-hand side are N2 results. As you can see, there's more frequent detection of N1 and N2 in the solid samples as compared to the paired influent samples. In addition, the solid samples achieve much higher concentrations than the influent samples that were quantifiable, on an order of magnitude of about 1,000 times higher. And this data was all generated using dd-RT-PCR. So in response to this data, we felt comfortable focusing solely on primary subtle solid samples, moving forward with our wastewater-based epidemiology project. At this point, we wanted to process high-frequency samples to create a longitudinal time series from one plant in San Jose, California. And the results from this are shown on the right-hand side. In the top panel are the number of confirmed cases in the sewer shed from this plant in San Jose, California. We had a bump we had our first wave of cases in March. We saw a decrease in cases and then a rising number of cases again at the end of June, beginning of July. In the middle plot are the concentrations of N1 and N2 in copies per gram. And as you can see, we see an initially high concentration of N1, the black boxes, and N2, the red squares, followed by a decrease in concentrations, and then again an increase at the end of June, beginning of July. And so we can see that the clinical case data is reflected in our wastewater data. At this point, we wanted to normalize our data for any dilution factors that might be happening due to stormwater or industrial wastewater dilution. And so we normalized our data, our N1 and N2 data, by the concentration of PMMOV, which again, we use as a measure of fecal strength of the wastewater. And when we normalize, we can again see this U-curved shape, which is reflected in the unnormalized wastewater data, as well as the clinical case data. And interestingly, the concentrations from March almost matched the concentrations in July, which suggested to us that there may have been undertesting in March. And in fact, our county epidemiologist colleagues agree that testing was greatly underestimating cases at the beginning of the pandemic in this sewer shed. The results of our recovery controls for this high-frequency longitudinal time series are shown here. We were, again, using bovine coronavirus, a spiked-in vaccine, as a measure of our recovery. The percent recovery is shown on the y-axis, and as you can see over this time series, we have a wide range in recovery. And in the bottom plot, we can see our PMMOV concentrations in gene copies per gram on the y-axis. And you can see that we have some variability in PMMOV, but we can expect from this one wastewater treatment plant a fairly stable concentration of PMMOV within the plant. So how are we planning on using this data? We would like to be able to ID outbreaks or hotspots. We have several large cities where we have multiple wastewater treatment plants within that city, and we think that that data could be granular enough to be able to identify hotspots within a city. We'd like to be able to show increases in community spread, as you're able to show with the San Jose data, and also potentially show decreases in community spread. And potentially show that even if clinical data isn't showing any confirmed cases within a community, if we have detections for N1 or N2 in wastewater, then we could show that there is actually infections in the community. We have learned a lot along the way. We have many conversations about the following important topics that I'm sure everyone is talking about. The importance of controls, particularly blanks and negative controls. We run one extraction blank with every batch of extractions that we do. In addition, we've discussed using field blanks. We're currently not using field blanks, but we have talked about the ability of field blanks to detect any contamination in the sampling process. In addition, we've talked about the number of NTCs to run and how to conduct those NTCs for DD-RT-PCR, and we've tried to develop fairly rigorous QA/QC procedures to detect any contamination during the RT-PCR steps. In addition, inhibition controls. In our group, we use dilutions to control for inhibition. For every sample that is run for N1 or N2, we run the RNA extract either 1 to 10 diluted or undiluted. And in that way, we can assess any impacts of inhibition in each sample. There are other methods to use, but that is the one that we use. In terms of recovery, we are using bovine coronavirus, a spiked-in vaccine, to estimate % recoveries from dewatered solid samples. As I showed a few slides ago, we do see a large range from less than 1% to 24%, with a mean of about 4%. We have developed QA/QC procedures. We drop any samples that we drop any data that has recovery of less than 1%, and we re-extract and re-quantify those samples. And in addition, we've started using PMMOV not only as a fecal strength indicator, but also as gut check data. Because we can expect a fairly constant level of PMMOV from a sample within a wastewater treatment plant, if a sample has drastically lower concentrations, we go back and re-extract and re-quantify that sample to make sure that that low concentration is not an artifact of a problem with our laboratory processes. So the next steps for our project SCAN, we're going to continue to work with public health officials on the implementation of our data. We are going to continue to retrospectively test samples. We have samples from 50 wastewater treatment plants around the country, so we are just continually going back and retesting those samples. In addition, we have started a pilot project in collaboration with Verily, which is a Google Life Sciences company that has industrial lab capabilities. And we partnered with them in 8 wastewater treatment plants around the country to provide a 24-hour turnaround time on samples and provide data in a dashboard. We've submitted our preprint to medRxiv. The details are listed here if you'd like to see any details from what I've just spoken about, and our entire team is listed here. So with that, I am happy to take any questions.
Perfect timing, Katie. Thank you. Okay, so I will ask the questions that are upvoted. So we have time for a few. So the first question, Katie, is how do you quantify daily loads to a sewage treatment plant in full scale with the primary solid concentrations in your sample?
I'm sorry, I didn't hear the first part of this question.
Okay. It sounds almost like a test question.
Oh, okay.
How do you quantify daily loads, I presume of SARS-CoV-2, to a sewage treatment plant with your measurements? How would you do that?
Well, we do use, in terms of daily loads, we use pepper mottled mylovirus as a way to control for how much human-derived sewage or human-derived impact is in each sample. So we use PMMOV to normalize for the contributions to a sample by humans.
Okay. Another question is, how useful do you think bovine coronavirus is as a recovery control, since one of the results reported in the preprint is that even when recovery was 0 of bovine coronavirus, we were still able to measure PMMOV and the SARS virus, even when recovery was reportedly 0?
Yeah, that's a great question, and it's certainly confusing. I think that when we think about recovery and inhibition, I mean, we have found evidence that each target behaves differently. We see different inhibition between targets, different inhibition between days of samples. So there are definitely going to be some differences in recovery, even when you're using bovine coronavirus as a surrogate recovery target as compared to SARS-CoV-2. However, I still think it's useful and it's very important to include an enveloped virus as a recovery control. And I think we're still sort of sorting through any issues that we're having with that. But I mean, there are questions there, but I think it is useful to have it. That way we can flag any problems within our processing and re-extract and re-quantify any samples to ensure that if something is a non-detect for N1 and N2, it's actually because it's a non-detect, or at least it's below the limit of detection, and it's not a problem with our laboratory method.
Okay. How do you communicate your results to the health department in San Jose?
Yeah, that's a great question as well. So we have weekly meetings with Santa Clara County. And so every week we talk with them about the laboratory processing. We've talked with them about our experiments in the lab with the influent and the solid samples. And so we have weekly meetings with them, and we also email with them, and we make sure that they are up to date with the laboratory methods that we're using, and as well as any projects that we're working on, particularly the pilot project. And yeah, that's the way that we communicate with them.
Okay, we're out of time for questions. Thank you, Katy, for a great job. I know that there are unanswered questions here. And so, Katy, I know you want to listen to Xavier's talk, but if we can maybe try to answer some of them by typing the answer, or if you still have burning questions, please email Katy. So thank you, Katy. So Yesith has joined the panel. She's one of the co-PIs on the RCN grant, and she'll be doing the Q&A for Xavier for his talk. So I'd like to go ahead and hand it off to Xavier, who's a postdoc at EPFL. And thank you, Xavier, for joining us today. You can go ahead and share your screen.
One second. I don't find the PowerPoint.
Well, while you're looking, I'll just say, I wouldn't want to be the one asking my own student the questions, and I've tried really hard not to answer the questions myself, because this is Katy's talk. But unfortunately, Kyle Bibby, who would normally maybe ask questions, had to teach, so he couldn't be here.
Okay. Now you see my screen, sorry.
Yes, it's great, Xavier. And so, Xavier, at 20 minutes, if you're still talking, I will turn on my video and I'll interrupt you, okay?
Good.
Okay, thanks.
Well, first of all, I would like to say thank you for the organizing committee to give me the opportunity to present our work that we have been conducting here in EPFL in Lausanne, and also in collaboration with the Aquatic Research Institute in Zurich. My presentation is called Pitfall in Measuring SARS coronavirus 2 in Sewage. And well, I'm gonna start saying that before coronavirus era, we had projects, everybody, that were slightly different. In concrete, I moved to Switzerland to work in a project that's called MetaVir, and basically was focused on the optimization and next generation sequencing techniques to study viral populations, and with the ultimate goal to use sewage as a public health surveillance tool. So all of you are aware that that sewage harbors a huge variety of viruses, and well, it's not new that sewage could be used to do environmental surveillance as a way to see what is circulating in the population. Particularly, I like a lot this study that was published this year, in which Bisex and collaborators show the power of sewage as an environmental surveillance tool by using amplicon sequencing and enterovirus. You can see in the upper panel that they monitored the sewage that was impacted in a particular region in France, and that they compare the enterovirus types detected in sewage with those that were reported in the clinical data. And what they found is that sewage was able to highlight a lot of enterovirus types that are silent and are circulating in the population, as you can see, because there are a much wide variety of enterovirus detected compared to the clinical data, but also they showed this particular red enterovirus corresponds to Coxsackie-virus B5, that the sewage was able to detect it much sooner, and in a large proportion, it became the majority enterovirus strain detected. And afterwards, after some weeks in the clinical data, it was the one that was causing more clinical outcome. Well, I like to show this study because for me, it shows a little bit the powerful of sewage as a tool to do environmental surveillance, and is the kind of research I think it's super cool, no? So in the context of the pandemics, we are wondering if actually SARS and sewage could be a good tool to monitor the impact of the SARS coronavirus 2. What we know right now is that just the clinical reporting is not enough to identify the level of impact of a particular pandemic. In concrete, in the United States, as during the first wave, and as the last data was April 18, it was shown that the clinical cases could be 3 to 20 times higher than actually the confirmed cases. So in that sense, in this context, sewage could be a tool that we could use to estimate the real incidence of a particular disease. It sounds a little bit weird at the beginning because SARS was primarily associated with respiratory problems, and respiratory viruses and environmental surveillance is nothing, for sewage is nothing that has been much explored. But there is some data that supports that maybe it could be a good tool to be used. In this sense, we know that, as Katy also showed before, half of the patients shed viruses, shed the SARS coronavirus 2, symptomatic or asymptomatic, and this shedding time can be as long by average as 12.5 days. Plus, despite some variability in the load of the virus shedded, it can even sometimes reach 10 to the 7 genomic copies per milliliter. So it seems that it could be possible to apply the environmental surveillance using sewage to track the pandemics. So we decided to start exploring this field of wastewater-based epidemiology and SARS coronavirus 2 in Switzerland. And we are currently working in several projects, which I'm just gonna show a little bit now. The first project that we have been working on, it's the first wave time course. So for this first time wave, what we did is we select 3 wastewater treatment plants, one in Zurich. For those who doesn't know Switzerland, Zurich is the biggest city in the country. Then we have also selected Lausanne, which is the city where I'm based, and a region in Lugano, as a wastewater treatment plant that is placed in Lugano. And for this is most of that I'm gonna present today, it's related to the first wave time course that we have been following from early March to the end of May. Then we also have a project, because in the meantime, we have also been collecting samples from this region in Lugano in the map. I make it bigger here. And you can see that this region has 9 green spots that they correspond to the wastewater treatment plants that we are sampling. We are interested in this particular region because it borders with Italy here. And as you might know, Italy has been one of the most striked places in Europe during the first wave. And also, it's important to highlight that we think that this part of the country might be one of the main entry points of the virus within the country during the first wave, because around 80,000 workers cross daily the border between Switzerland and Italy just to work. So they go, they come, they work, and they come back home. So this has been happening during all the pandemic. And we believe it's interesting to do even a phylogenetic analysis of the kind of SARS and the strains that were circulating during this time at this region. And then we have a third project that is now currently ongoing, that is the real-time monitoring, which we narrowed down to 2 cities, Lausanne and Zurich. So how are we handling the sampling and processing of our samples? So as also Katy highlighted, we have been working under a lot of pressure and with a lot of constrictions because the lockdown, the labs have been closed. We have been having supply issues. So at the beginning, we couldn't process in real time the samples. So we had to storage them. The type of samples we have been storage them in the freezer, which is important by something I'm gonna explain afterwards. So yeah, we have been collecting 50 milliliter 24-hour composite samples from the different wastewater treatment plants, almost daily, but sometimes it's one day yes, one day no. And what we have been doing is treating them as follows. No, we just filter them by 0.22 to remove particles and bacteria, and the filtrate we put it in a centricon. That a lot of us are using to concentrate the samples, and it's based on ultrafiltration. By applying this method, we are concentrating approximately 250-fold the viral particles. This viral concentrate has then been extracted using Qiagen RNA viral kit, and then inhibitors have been removed by using an exclusion column system. The nucleic acid extraction has been quantified by using real-time qPCR for 2 targets that involve SARS and 1 and 2 genes based on the CDC assay. So in total now, we have analyzed for Zurich 35 days from March to the end of May. And from Lausanne, we have been analyzing 38 days from the same dates. And here I'm gonna present in this slide the first wave, how it looks in Zurich. So as you in black, you can see represented the number of average reported cases in the canton of Zurich. And in this y-axis, you can see the number of genomic copies per milliliter for both targets, N1 and N2. And you can see that somehow the number of genomic copies, it fits under the reported cases for the particular canton. I want to highlight that in this case, the number of reported cases and also the quantifications are low compared to the next scenario that I'm going to show you. This is because Zurich was particularly not heavily affected compared to Lausanne during the first wave. So also the reported cases are lower and the genomic copies per reaction are also lower. When we see the first wave, this is the representation of the first wave in Lausanne, we can see that both the number of reported cases and the number of genomic copies per reaction are higher compared to Zurich. This matches because, as I told you, we have a high number of cases in this canton. And we can see that also the number of genomic copies follows within the curve of the reported cases. And then in the 16th of March, Switzerland applied a soft lockdown, which clearly affected the number of cases because it drastically reduces the number of cases since then, but also affected our quantifications in sewage, which decreased it. And when we arrive in May, basically, we just could detect at the beginning of May, and then all of the samples have been under-detected. So it seems that we had a method that worked. But we wanted also to see if the method worked for different case scenarios, no? Because I showed you two different curves. And what we did is to correct the reported genomic copies per reaction according to the water discharge for each site. And when we plot this viral load against the clinical reported cases, we see a correlation between, in both sides, no? Between the clinical cases and the viral load reported. This is interesting because it happens that despite Zurich had a lower number of cases, it also had the double the water discharge. So if we didn't correct this, we couldn't see this correlation. Despite this apparently correlation, no, we know that the data is still scattered and the results are promising, but maybe some improvement or refinement needs to be done in the method so we get better quality data. In that sense, we have been analyzing our current pipeline and we have tried to identify potential pitfalls that might be responsible for this maybe not perfect matching, no, that we observe. So at each step, that can be sources that might introduce bias or affect our representation of the data. Since the storage protocol and duration of this storage, because as I told you, a lot of our samples have been in the freezer for some months, and this may have affected the quantification of the virus. But also, we know that during the filtration and concentration steps, there might be losses that might be related to the association of the virus to the organic matter in suspension, or losses during the concentration step itself. That people that has been working on viral environmental virology knows that concentration is the weak point of all of these studies a lot of times. Then we have been, then some of the losses can be also related to RNA extraction because it's not 100% efficient. And also we apply some inhibitor removal treatment, which might also affect the quantifications. Finally, PCR inhibition is a known phenomenon and it can also be responsible. So we have identified all these points, and also we have implemented some kind of measures that try to minimize the impact of this variability or these potential sources of losses of the virus. For example, we always analyze our samples by duplicate biological replicates. And also to tackle a little bit the part of the concentration and extraction, we spike our samples with different surrogates, which I might talk about them afterwards. We also measured pepper mile mottle virus, which is a plant virus that seems to be present in constant concentrations in sewage. And an important change in this measurement could be indicative of a dilution of the sample because it was more rainy events before that sampling date, or because something happened during the lab work. Finally, to deal with inhibition, we also spike our viral concentrates with a known concentration of SARS coronavirus, and we measure the degree of inhibition on the sample. I'm going to focus right now on what happens on the storage, because as I told you, we have had our samples for some time in the freezer, and we want to know how much this storage thing can affect our quantification. For this reason, we have designed an experiment in where we stored 50 milliliter aliquots of the same sample in different storage conditions. We have froze directly the sample without concentration. We have stored the viral concentrate, and we have also stored the RNA and extracts. And we have done this under two different temperatures, in the fridge and in the freezer at -20, and for several times, like one week in these conditions and 1 month in these conditions. And this is preliminary data because this experiment is currently ongoing. But what I can show you is that the reference in blue, which was the sample directly processed the same day, all of it, and measured by qPCR, show a slightly higher quantification that the same wastewater stored at -20 for a week. When we analyze the concentration of N1, because this is N1 data, N1 of the concentrate at -20, the extract at -20, and the sample without freezing at 4 degrees, we see that the quantifications are higher than the reference. So we can assume that in any case, if there is a loss, this loss might be done during the storage of the sample. However, as I mentioned, this is an ongoing experiment and we need more time, and we expect that with the 1 month results, we will have clear outcome if this is really a decay on the concentration of the virus or is within the error of the measurement of the experiment. Another point is, as I told you, the steps that involve the filtration, the concentration, and extraction. This could also represent a potential pitfall. And to tackle this, it brings me to the use of surrogates. In this project, we have, since the beginning, spiked with 3 envelope viral surrogates at the same concentration, approximately. All of them are envelope viruses, and some of them are well known for all of you, like murine coronavirus, because it has been used in several studies as a surrogate for the SARS coronavirus 2, because it belongs to the same family. So we assume that the behavior of this virus should be similar to the one of SARS, no? Of the actual SARS we are tracking in sewage. Then we also have been using the Phi bacteriophage, which is a Pseudomonas bacteriophage, which has a total different conformation of the genome because it's double-strand RNA and segmented. But this has as a side effect that it's easy to grow and easy to culture in labs that not necessarily have to be P2. And finally, we have another surrogate that it's Sendai, for which we have developed a specific real-time qPCR assay that belongs to the paramyxovirus family, and it's the genus Respirovirus, which includes other important human pathogens such as human parainfluenza or respiratory syncytial virus. And I was talking about the pitfall because some studies have suggested that a lot of, 2 envelope viruses tend to be more attached to organic matter compared to non-envelope viruses. And this data, despite it might seem low, it's worrisome when you think that it's only based on infectivity. So if you go at the molecular level, we would expect higher quantifications. So in order to see the involvement or how this envelope virus behave, and to tackle the possible pitfall of losing them during the filtration, we made an experiment and where we collected the same wastewater and we tried to separate the 2 fractions. The liquid fraction by applying centrifugation. The liquid fraction we analyzed by using the centricons, as mentioned before, ultrafiltration, and the solid phase was extracted by using a magnetic extraction kit. And when we observe the total recovered viruses, we can see that most of the total, about the overall recovered viruses, most of the envelope viruses are actually found in the pellet, more than in the liquid fraction, especially for the 2 surrogates that are animal viruses, murine hepatitis virus and Sendai virus. For the bacteriophage Phi, we see a behavior that is half between the pellet and the liquid. When we focus on the liquid phase of the samples we have been processing, the result doesn't change very much because we observe that the recoveries are low, usually below the 10%, but also they are highly variable between samples. So they don't show like a similar recovery rate, even though samples are processed the same way. So we observe poor and variable recoveries in the liquid phase. But also when we try to see if there is a similar behavior between the surrogates themselves, we see that there is no evident correlation between them. So this is the pipeline that we have been using for our processing the samples. And even though, so as I have been explaining, we have been using several ways to try to refine and produce consistent data. We have been using surrogates, we have been using replicates, we are using pepper-mile mottle virus to try to normalize the quantifications, and we are attending for the inhibition. And what happens when we try to include this information on the raw data that we have? This is my last slide, Ali, so I think I'm on time. So in this graph, I try to represent the raw data of N1 quantification per microliter with the actual flow rate. And what we can see is that the trend is similar. They follow the similar curve pattern. So I wouldn't expect a much significant change in the data overall. When we do the same approximation by pepper mile, we see that the curves follows the similar pattern in a similar way. But when we analyze this information by normalizing of one of the surrogates, what we see is that the curve follows a different pattern, which is not surprising considering the low recoveries and the variability between the samples. So this is just because, as all of you, we are trying to understand how to better represent this data and how to give biological significance of the data we are producing. So this is just preliminary information that we are still working on it. So as I mentioned, there are several potential pitfalls related to the storage. Maybe freezing is not the best way to storage these samples. When we are considering only the liquid or the solid fraction, we are selecting one part of the story, and both of them have pros and cons. For example, the liquid fraction could be more representative because we are knowing exactly which sample day we are testing. And also we can normalize the data to compare different sites, as I showed before with Zurich and Lausanne, but also had some minor bad things. Like most of the viruses, we know they seem to be associated with particles, so we could expect lower quantifications. And also, particles could be more useful during not peak zones of the current pandemics. And then the role of the surrogates, as Katy also showed, is a little bit, needs much further interpretation and work to understand this. I would like to acknowledge all the people that has been involved during the collection of the samples, because it has been a hard work. All the people at EVAC that has been working in this project with us, and especially to the people at EPFL, Alex and Federica, which helped me a lot with this presentation. Thank you very much.
Thank you. That was great. So we have some questions from the audience. So for the liquid solids comparison of the surrogate, those are out of 100%. And so one question was if the fractions or if the sum of the liquid and solids always equaled what you spiked in, or what were the losses associated with each portion?
I don't think I understand the question. Can you repeat or reformulate, sorry?
Yeah, so I think it was based on a specific slide. You had liquid and solids comparison of the...
Can I share just a second slide? And because I unshare, I think...
yeah.
Yeah, I know which slide do you mean. Huh? This slide represented the... it was normalized by the total recovered virus. So actually we have another slide where we show that most of the virus disappeared. We couldn't find it. So this slide is like assuming that the number that we have is 100%, how much percentage goes to each fraction.
Okay, so it's normalized to that, not total you added, but total you obtained. That was the question. So it's normalized to not what you added, but what you recovered.
Hello, I think I lost the connection.
Can you hear me now?
Yes, I lost the connection, sorry.
Okay. So it was normalized to how much you added, or it was not normalized to how much you added, it was normalized to how much you recovered.
Yes, exactly.
Okay.
Of the total recovered viruses, then we just assumed that it was 100%, and then we divided, because if we did it by the total spiked amount, then what we found out is that a lot of the virus was simply nowhere, which is not possible.
Right. Another question was, you had a step for inhibitor removal prior to qPCR, and the question was, I guess you talked about testing for inhibition by spiking in the SARS-CoV-2, and so the question was, you know, did you realize that that step was definitely necessary, and what are your results for qPCR inhibition?
Okay, so we have two different things. Okay, the inhibition test is performed on the extract spiked with a known concentration of SARS coronavirus 2, and then we compare the different CQ between water and the extract. And this data we still need to analyze in detail, because it was just produced last week, so we didn't have time to analyze it. So I cannot produce more insight on this. For the inhibitor column, so we have a column that is based on size exclusion. So in theory, the inhibitors should get trapped in the column, while the virus RNA should be extracted. And for this one, what we saw at the beginning is that for smaller volumes, we couldn't quantify if we didn't use those. You have to think that also these results were based in samples that were collected in May. So, and based on the SARS coronavirus quantification, because as Katy already highlighted, the inhibition affects much differently depending on the PCR assay. So we cannot use the results for enterovirus or for peppermile to extrapolate the inhibition, because it totally goes different. So it was based on May results, which had very low numbers of SARS here. And it was basically, we could detect or we couldn't detect. And when we increased the volume, in order to see that much volume could provide us more positive results, without the column, we couldn't quantify, we just couldn't provide, we could provide a qualitative result, but we couldn't give a quantitative result because it was outside of the quantitative region of the qPCR. But we definitely need to do a spiking experiment to assess the losses also by using this column, because definitely there are.
Well, thank you, Xavier. We're out of time, so we're gonna close the webinar. Thank you everybody for attending. And if you have additional questions for Xavier, I'm sure he'd be happy to answer them, and Katy as well. Great job, you guys. Very impressive and hard work. Thank you so much. And for all the attendees, we have another webinar in a few weeks, and I'll be sending out reminders. And if you have a student or postdoc that is working on this topic that you think would be a good candidate for the webinar, please let one of us know, Yeseth or me or Kyle Bibby or Ralph Halden. Okay, thank you everyone. Have a great day. Bye-bye.
Bye-bye.